Hi everyone, this is Miss Lasar, and today I'm going to be teaching you about mutations and gene expression. So take a moment to check out our amazing meme of the week, and let's talk about the questions that we're going to answer today. Question 1, how do our cells all have the same DNA, but look so different? Question 2, what happens when there is a mutation in DNA? And question 3, how can changes in gene expression and mutations cause cancer? During the lecture, do your usual good move of taking detailed handwritten notes using the template that your teacher has distributed, and just remember, write down all the details of today's lecture. So we're ready for our first question. How do our cells all have the same DNA but look so different? Here are some classic body cells: nerve cells, heart cells, liver cells, bone cells, intestinal cells. You have all of these different cell types. And even though they look and they act so different, they all have the exact same DNA. Every cell in your body has the exact same set of DNA. How's that possible? Well, these cells are so different, but what's different about them? It's the proteins that are in each cell. So now's a good time to remind yourself of the connection between DNA and proteins. Remember that a gene is a piece or a section of DNA that has the instructions for how to make a protein. And you have tons of DNA coiled up inside the nucleus of your cells. And that set of DNA, that's your genome. And that is all of the genes, all the instructions to make all the proteins that are in your cells. So using those vocabulary words a little more specifically, I might be talking about hemoglobin, which is a protein, so not DNA, but a protein that carries oxygen around the body inside of red blood cells. I could also talk about the hemoglobin gene, which is a piece of DNA with the instructions for how to make hemoglobin. And I could talk about the hemoglobin mRNA transcript. That is in purple in the lower left. That is a piece of mRNA that's complementary to the hemoglobin gene. So all three things on the screen here have the same information, but in different forms. Hemoglobin is the functional protein, the functional tool that can do a job. The hemoglobin gene is the instructions to make hemoglobin written in DNA. And the hemoglobin mRNA transcript is that same set of instructions, but written in mRNA, so that we can take it out into the cytoplasm, get it to a ribosome, and get it translated and turned into functional protein. So I've got a new term for you: gene expression. Gene expression is just another way of saying protein synthesis. We say a gene is expressed if a protein is made. So that means we have our gene, we rewrite it, we transcribe it into mRNA, and then we translate that mRNA into protein. We've expressed or made the protein. We say a gene is repressed if the protein is not made. So we don't transcribe it into mRNA, or we don't translate it into protein. Here's how to use these terms correctly in a sentence. I could say the cell is expressing the protein, the cell is expressing hemoglobin, or I could say the cell is expressing the gene for this protein, the cell is expressing the gene for hemoglobin. If I was using repressing, if I'm not making a protein, I would say the cell is repressing the protein, the cell is repressing hemoglobin. Or I could say the cell is repressing the gene for this protein. The cell is repressing the gene for hemoglobin. And both of those mean the same thing. So now let's look at those same cell types and look at what each cell expresses. Look at the proteins that each cell makes, starting with bone cells. Bone cells are hard, they're strong, they give your bones structural support and stability, and they can do that because they're surrounded by all of these rings of fibers that you can see in the picture. Those fibers are a particular protein called integrin. This is a protein that forms very hard fibers and gives bone cells its strength. We would say that bone cells express integrin. But what about an intestinal cell? This is a soft, squishy cell, doesn't need to be hard, doesn't need to make integrin. It does need to make a protein called lactase, which is a protein that breaks down lactose in food and allows your body cells to use it for energy. So take a look at the pink lactase that's able to grab onto lactose, that circle plus hexagon combined, and break it into two pieces, the circle, which is glucose, and the hexagon, which is galactose. We would say that intestinal cells express lactase, which means intestinal cells make the lactase protein. And bone cells, they don't make lactase. So bone cells repress lactase. Intestinal cells don't make integrin. So intestinal cells repress integrin or repress the gene for integrin. Let's look at lactase in a little more detail because this is a really good example of a gene that is expressed sometimes and repressed at other times. Everyone expresses the gene for lactase at birth so that all humans can drink breast milk because there's a lot of lactose in breast milk. Some people continue expressing the gene for lactase for their entire life, and so they can always drink milk of any variety and break down the lactose that's in that milk and use it for energy. But some people repress the gene for lactase when they turn usually just after infancy. And that means that they are no longer making the lactase protein and they can't process lactose anymore. You may have heard of these people. You may be one of these people because these people are lactose intolerant. They can't tolerate eating foods with lactose in them. And so that's an example of when a gene would be expressed at some times during infancy and repressed at other times, like during adulthood. So what actually controls gene expression? This is actually a really complex process and one area that scientists are constantly learning more about. I'm going to give you two examples of what controls gene expression. The first example is something called epigenetics, and these are physical changes in the DNA structure that prevent RNA polymerase from accessing a gene and being able to transcribe it. On the top, I've got DNA that's too tightly wound. It's so tightly wound and curled up that RNA polymerase, which we need to rewrite or transcribe the DNA into RNA, it's just too tight. RNA polymerase cannot get in to transcribe the gene. On the bottom, I've got DNA that's much looser, and RNA polymerase can easily get in between those spirals and can transcribe a gene. And so DNA on the bottom is a gene where transcription would be possible, and so we could express that gene. The gene on the top is one that would be repressed. Here's another example of something that can control gene expression. It's called a transcription factor. These are molecules that bind to DNA and are required for RNA polymerase to start transcribing a gene. Last week, some of you asked me, how does RNA polymerase know where to start transcribing or even which strand to start transcribing for transcription? And the answer is transcription factors will tell it exactly where to attach and where to start transcribing. You can see in the image that there's a blue transcription factor that's attached to the DNA as well. And if that transcription factor isn't there, RNA polymerase won't even attach and the gene will be repressed. If the transcription factor is there, RNA polymerase will be able to attach, it will be able to start transcribing, and the gene will be expressed. So on the top, our transcription factor is present, transcription will happen, and the gene is expressed. On the bottom, our transcription factor is absent, and so no transcription will happen, and the gene is repressed. Now we're ready for question 2. What happens when there is a mutation in DNA? A change in the DNA results in a change in the mRNA, which could result in a change to the final protein. A change in DNA is what we refer to as a mutation. And so, as you can already start to infer, there will be different effects based on the types of mutations that we see. We assign mutations into 3 categories based on their DNA change. So on the top, what you can see is a string of DNA letters, AAA, ATG, CTT, CTC. So they're broken up into codons already. Underneath that, you can see the complementary mRNA bases. And then underneath that, underneath each codon, you can see the amino acids. So UUU codes for the amino acid phenylalanine, which is abbreviated PHE. The first category of mutations based on DNA change is a substitution. So notice what's different in the DNA from the original strand, which is at the very top. In a substitution, one or more bases is changed or swapped out. So notice that in the third codon, the first C has been changed to a T. We've substituted a T for that C. An insertion is another type of mutation. We can actually stick extra letters in accidentally to our DNA. For example, you can see that in our insertion example, an extra C has been added before the original C that was in the third codon. So now, instead of going CTT, CTC, we go CC, TTC, etc. The third category of mutation is a deletion. So just like we can add bases, we can also remove them or delete them from the genome. And again, you can see our example. In our example of a deletion, we removed an A. So we removed an adenine. And what you might be noticing is that there's some different effects on the amino acids. In all of these cases, we ended up with a change in the amino acid sequence. Now let's talk about that. So we divide up our categories of mutations based on different things. First, the change to the DNA. Did we substitute a letter? Did we insert some letters? Did we delete some letters? But then we also have to look at the outcome of the mutations, which is the change to the amino acids. And again, we've got different categories, 3 different categories here. In our first example, we have a silent mutation. And it's called a silent mutation because even though we had a change in our DNA, we did not have a change in our amino acid. Our amino acids are the same as the original. So notice in our original DNA on the left, our DNA is ATG, that codes for UAC in mRNA, and that codes for the amino acid tyrosine. In our first change, the final G was changed to an A, but that UAU codon still codes for tyrosine. So we still have the tyrosine amino acid that we had at the start. That's a silent mutation. Our second example, the final G was changed to a C, and that resulted in a stop codon. UAG is a stop codon, which means we're going to stop translation early. That's called a nonsense mutation. A nonsense mutation is an early stop codon. And our final example, we changed the first letter of the codon, A, to G, and so that resulted in CAC in the mRNA and a histidine amino acid. So we actually just changed our amino acid. That's a missense mutation, and that's when one or more amino acids are changed. You might have noticed what type of DNA change we saw in all 3 examples. All 3 examples were substitutions. So we had a substitution that resulted in a silent mutation, a substitution that resulted in a nonsense mutation, and a substitution that resulted in a missense mutation. You could say a silent substitution, a nonsense substitution, or a missense substitution. Let's talk about silent mutations in a little more detail. Let's look at proline. So if we look at our codon chart, we can see that there's 4 different codons that all code for proline: CCU, CCC, CCA, and CCG. That means if in our DNA we change CCU, well, we'd be talking about our mRNA. If we change CCU to CCC or to CCA or CCG, we're still going to get a proline. Silent mutations are pretty much only seen in substitution. So that's just a clue for you. If you see a silent mutation, it's almost always caused by a substitution. Nonsense mutations, stopping the transcript early because we have an early stop codon, these are devastating. If we're only getting partway through our protein code, if we're only getting some of our amino acids into that amino acid chain, we are certainly not going to make a functional protein. Nonsense mutations are typically disastrous for the protein function. Missense mutations, on the other hand, can have a major effect or a minor effect, and it's kind of hard to predict what the effect will be. If we're just changing one amino acid, like we change a glutamine to an aspartic acid, like you can see in the example, that's just one amino acid out of hundreds. It may have a very minor effect, or it may completely destroy the function of the protein. It's pretty hard to predict. There's one other type of mutation we got to talk about, and that's a frameshift mutation. So let me back up and talk about codons before we talk about what a frameshift mutation is. On the screen, you can see I've got 3 strings of mRNA. and they're the exact same string, CAG, repeating over and over and over. And so I've already asked you to, in an earlier lecture, divide up these letters into codons, three base chunks. Well, most students would immediately go like this and divide up all the letters into three base chunks. But why do you have to do it like that? Why couldn't you divide up the letters like this or like this and just put your first codon at a slightly different location? Notice that if we change where we start determining our codons by just a letter, the codons themselves change a lot. Like in our first line, we've got CAG repeating over and over. In our second line, we've got AGC repeating over and over. And in our third line, we've got GCA repeating over and over. What we're looking at right now is the reading frame. This is how you divide the codons. And what determines the reading frame? The location of the start codon. The location of the start codon determines the reading frame. You know that the first step of translation is find the start codon, the first AUG that you see. And then after you found the start codon, then you'll divide up the remaining chunks, the remaining letters into three base chunks. And then you'll use your codon chart and you'll translate it, and we would get something like this. AUG codes for methionine, CAG codes for glutamine, and then we'll have glutamine repeating over and over and over and over. But take a look at the second line. In the second line, I put the AUG just one letter in. So it's the exact same sequence. I just put the AUG one letter in, and now I've located my start codon. I divide up the remaining letters into three base chunks, codons, and I'll translate it. And now I get methionine and then serine repeating over and over and over and over. These would be completely different proteins. Instead of glutamine repeating over and over, I've got serine repeating over and over, a completely different amino acid. And my third line, I put my start codon just two bases in. So I'll divide up the remaining letters into three base chunks, the codons, and translate it. And now I have, again, a completely different sequence. Methionine as my first amino acid, as it always is, and then alanine repeating over and over and over. And so what you might be figuring out and inferring is that mutations can change the reading frame. Let me show you some examples. Here is our original string. We go methionine and then glutamine over and over and over and over. But what if I put an extra U in there? And instead of having my normal codons, now everything's kind of shifted over by one because I inserted an extra U. Now I'm going to have a methionine, then I'll have a serine, and now I have a bunch of alanines over and over and over. Notice how these two amino acid sequences are just completely different. By the way, I'm noticing this like weird UUU on the top line. I'm sorry, that is a typo. If you can ignore that, please ignore that. Now let's take a look at another type of mutation that can have an effect. So we've got our normal sequence on the top. We've got our sequence with an insertion mutation in the middle. And now here's our third sequence. This is a deletion mutation. I've deleted two bases. I got rid of the C and the A that were right after the AUG. So same deal, I locate my start codon, I divide up my letters into three base chunks, and I translate them, and now same deal. I have a major change. Instead of methionine with a bunch of glutamines after, I have methionine with a bunch of alanines after. So how bad are these changes? Well, let's talk about a protein and its function. A protein's function is determined entirely by its shape, just like a tool. So think about a tool like a cheese grater or a watering can. If the shape of the watering can were changed very slightly so that the spout pointed back into the can, it's a very slight change, but think about its function. It wouldn't work at all. You can't water plants with that watering can. And think about a cheese grater. If the shape of the cheese grater was changed so that it had a smooth outside, you could smash cheese against it, but it wouldn't grate. So the amino acid sequence determines the protein shape, and small changes in form can significantly change the function. Let's talk about those changes again. Well, if there's a small change, the protein may be partly functional. What do I mean by a small change? Well, what's in the box on top? A small change is a change where one or two amino acids have been changed. That's what we consider small. What about no change? Well, if there's no change, the protein will be fully functional. No change would result from a silent mutation where we've got a change in the DNA that doesn't change the final amino acid sequence. It would also result from a mutation before the start codon or a mutation after the stop codon. If it's outside of where we're translating, it's not going to be a big deal. What about a moderate to large change? That is a change of 3 amino acids or more. Also, early stop codons. Both of those count as moderate to large changes. And in those cases, the protein will be non-functional. You can pretty well assume that with 3 or more amino acids changed or an early stop codon where we're just missing a chunk of the protein will not be functional. So you're going to be presented with mutation problems on paper. And what they'll probably be like is saying, here's a DNA sequence, here's the mutation, what is the impact on the final amino acid sequence, and what kind of mutation are we looking at? So here's how to solve those problems. Step 1, identify the type of change made to the original DNA sequence. Was it an insertion, was it a deletion, or was it a substitution? Write that down. That's the first part of your answer to what type of mutation we're looking at. Step 2, transcribe from DNA to mRNA, find the start codon, and divide the bases into codons. Ask yourself, was there a shift in the reading frame from the original DNA sequence? If the answer is yes, you're looking at a frame shift mutation, and you should just finish your translation to see what effect that'll have on the amino acid sequence. If the answer is no, and there's not a major shift in the reading frame, then finish your translation and compare your translation to the original translation. Ask yourself, was there no change from the original amino acids? That's a silent mutation then. Were some amino acids changed? That's a missense mutation. Was there an early stop codon? That's a nonsense mutation. So you'll have a 2-part answer. I'm looking at a missense insertion. I'm looking at a silent substitution. I'm looking at a nonsense deletion. I'm looking at a substitution that resulted in a frame shift. Every answer to what kind of mutation are we looking at will have 2 parts to it. What was the change to the original DNA sequence, and what was the change to the final amino acid sequence? Feel free to recreate this flowchart in your notes. It may be helpful. Let's look at an example of how mutations can affect a protein's function. A classic example is sickle cell anemia, which you've already learned about. In sickle cell anemia, a mutation in the hemoglobin gene, it's a single substitution mutation, causes a single glutamine to be changed to a valine. So this is a missense substitution. This results in a sort of functional protein, right? That kind of fits. We've had one amino acid being changed, and we know that if there's 1 or 2 amino acids that are changed, we anticipate the protein to be sort of functional. This sort of functional hemoglobin has a new shape, and the new shape of the hemoglobin causes it to build up in long strands inside red blood cells. And as you learned about earlier this year, red blood cells with the mutated protein have a long, skinny sickle shape, and they get caught in capillaries. They just do not deliver blood and oxygen as well. This results in symptoms like pain from loss of oxygen to those vital organs and fatigue from not getting enough oxygen body-wide.